everybody. Good morning, good afternoon, good evening. I'm Ian Coates, and welcome to week 6 of Biology 1021, where today we're going to be talking about mitosis and meiosis. Now, these two words, they sound very similar, but they are not. They both pertain to reproduction, cells growing and developing into something new, but their pathways are quite different. And that's what we're going to focus on there today. We're going to look at some cancer, different forms, what exactly cancer is, and how to prevent it. All right? So to no further ado, we're going to continue, but just please make sure that we've just had our test 1, and now we're moving on into test 2 material. Okay, here we go. Now, prokaryotic reproduction, here we're talking about bacterium, right? And you know, they have circular DNA, which is quite different from ours. Okay, and they have a starting point. So start, copying, 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 stop. How do you know where you stopped? Well, that's where you started. Their DNA is very primitive. They've also got these plasmids as well. Okay, they're involved in antibiotic resistance. Just a little bit of a recap there, because this is where we're going to go. So, cell division in prokaryotes takes place in 2 stages, which together make up a simple cell cycle. First of all, number 1, copying of the DNA. This process is called replication. Okay, as I said, copying the DNA, starting is the exact same spot as the stopping position, so it's very simple, not like ours. Okay. Number 2, split the cell into 2 to form 2 daughter cells. This process is called binary fission. Okay, we'll hit that there in a second. All right. So the whole idea here that the hereditary information in a prokaryote is stored in the DNA, same as any other organism. The prokaryotic chromosome is a single circle of DNA. DNA replication begins with the unzipping of the double-stranded DNA at a point called the origin of replication. Okay. A new double helix is formed by adding complementary nucleotides to the exposed DNA strands that have been unzipped. Okay, so the DNA opens up, we're able to make copy of it. That's how it goes in bacterial reproduction. So after the DNA has been replicated, the cell grows and then splits into 2. This process is binary fission. So you have a look here. We can just go step by step. Okay, and you'll see here that here's your normal bacteria here. Okay, and origin of replication, starting, stopping, and you'll see there the DNA is being copied. 2 copies are created. They move to either side, where telekinesis begins, the whole idea of pinching off of the 2 cells because the cell has elongated and got bigger, and then we have the end product. Okay, lots of good stuff there. Okay, and even reading in there as well. Bacteria use other mechanisms for increasing genetic variation. Now you remember there, genetic variation is the very fact that 2 parents can have 4 different children and they all look completely different. Their DNA is different, their ability to fight off diseases is different. Okay, and that's the whole point, right? Because if there's one disease that will eliminate one of the offspring, we do not want it to eliminate all of them. Okay. Eukaryotic organisms have sexual reproduction. Bacteria use the following reproductive mechanisms. There's conjugation, which we've heard of already. Remember the sex pili. There is transformation, transduction, and transposition. Transposition, okay, and the movies here, these are great. Please pause the movie or please pause the video there and watch these movies. They're good for you. So transposition, this is where bacteria naturally move genomic DNA back and forth to and from the plasmid. Okay, so you see there's your DNA, sorry, just a shot there, DNA in the plasmid right here. So not only to have this versatile little DNA that can be moved and shuttled between different bacteria, we can move information back and forth from the genomic to the plasmid. Okay, quite interesting. Okay, so that was transposition, moving DNA back and forth between the genomic and the plasmid DNA. Okay, there is conjugation, which we've already spoken about there last week. Using the sex pili of one bacterium is able to exchange information with another bacterium, not even of the same species, doesn't have to be. Transformation. Bacteria can undergo this process when they incorporate DNA from their environment into their own genome. So, scientists there, if they want a certain bacteria to pick up some type of DNA, all they have to do is put it in the medium that the bacteria are living in, and they will pick up that DNA and make it part of their own. Incredible, isn't it? Transformation. And the last way of genetic material exchange is called transduction. And this is by using, remember that bacteriophage, a virus that only infects bacteria, is able to transmit viral DNA from one bacterial cell to another. And this process here is called transduction. Okay? Transposition, conjugation, transformation, transduction. Okay? And you'll see here we have conjugation, transduction, and transformation, the whole idea of what is happening. Okay? There's your A, B, and C. This is transduction. Okay? Right here we have B. Oh, sorry. That's your sex pili. I was looking at that going, is that a sex pili or not? So yes, this is your conjugation, the whole idea of moving information there via the sex pili. You have transduction here. This is where the bacteriophage is injecting its information. Okay, and then we have transformation. DNA fragments there from donor cells or just found in the environment. The bacteria is able to pick it up. Okay, good stuff. Eukaryotic cell cycle contained the eukaryotic cells, since they contain more DNA than prokaryotic cells, okay, and the DNA is also packaged very differently. DNA in eukaryotic cells is linear, it's not circular as in prokaryotic, and it's packaged into these compact chromosomes. Okay, how many chromosomes, DNA macromolecules, do humans have? Lots. Okay, how many chromosomes? 23 and 23. The eukaryotic cell undergo two different mechanisms there to divide the DNA. There is mitosis. This is what's going on right now in the cells in your arm, your leg, okay? Somatic cells are body cells. These are just cells there. When a cell gets a little older, it needs to divide, okay? And you want two exact copies of the previous mother cell. The mother cell will divide and create two daughter cells that are exactly the same. Somatic cells, body, mitosis, the end result is the same as what was there earlier. Meiosis, not so. Here we're only talking germ cells. Germ cells, sex cells. Egg cell, sperm cell. Okay? A body cell has 46 chromosomes. 23 came from the mother, 23 came from the father. Okay? And so the sex cells only contain 23. Okay? That's what we... And meiosis allows for each one of these sex cells to be different. Okay? So, the eukaryotic cell cycle is divided into distinct phases. You'll see what's going on here. Okay? You have, this is all called interphase. Okay? This orange part of the circle here. Okay, and interphase is composed of what? The G1, the S, and the G2 phase. Okay, G1, period of cell growth before the DNA replicates. S phase is a period when DNA replicates and the chromosomal proteins are duplicated. Okay, so, and then we get into the G2 phase of interphase. This is the period after DNA replicates, the cell prepares for division. Okay, so the DNA has already been doubled by the time mitosis actually begins. And you'll see there that mitosis, there are 4 phases to it. And if you can remember, PMAT, PMAT, okay? There's prophase is the first one, metaphase, anaphase, telophase, and then outside of mitosis there is cytokinesis. This is the pinching off of the 2 cells. We'll get more into this as we go on, okay? So, mitosis, homologous chromosomes contain the same genes, but they may have different variations of those genes. One could come from mother, one could come from father, which are called alleles. One homologous chromosome is obtained from each parent. Sister chromatids are exactly the same and are a result of DNA replication. Sister chromatids. Okay, good. This is what we call a karyotype. Now, it's not like these are all the chromosomes of a particular human being, okay? And you'll see there that you have 22 of them, and they're not all lined up. They've taken pictures of these and they've lined the pictures up. It's not like they tell the chromosomes, okay, everybody line up. All right, so you'll see 1 to 22, we call these there the autosomes, and then the 23rd is your sex chromosome, okay? And this is what we call a karyotype, which is an arrangement of the chromosomes that can be viewed, compared, and shared based on size, shape, and centromere location. Okay, the karyotype right shows 23 pairs of human chromosomes. Okay, so this is a pair of homologous chromosomes. One came from mom, one came from dad. Mom, dad, mom, dad. Okay. So, here's some chromosomal trivia. The number of chromosomes does not determine the complexity of the organism. Look at shrimp. They have 92 chromosomes. Potatoes. They have 2 more than us. How is that even possible? Right? But they don't... keep your nose high in the air. We're still top of the food chain, even though we don't have the greatest number of chromosomes. So the chromosomal tips, also called telomeres, okay? Every time a chromosome doubles and the DNA replicates, these telomeres, they get a little shorter, and these happen at the end. And these telomeres, once they get to a certain point, then this chromosome gets the chomp. So you can only multiply the DNA a certain number of times, okay, before the telomeres shorten to a point where it will not go any further. Okay. So, still talking about the chromosomes there, and now we're looking at genes within the chromosome. Now, these genes here, this is what there's gene 1 and gene 2. One came from father, one came from mother. Okay, and these provide the information there that we're looking for. So prior to cell division, each of the homologous chromosomes replicate, forming 2 identical copies called sister chromatids. So the DNA doubles, okay, these chromosomes double, and they make a copy of themselves. So there'll be 2 copies, 2 identical copies of every single chromosome. Okay, and those 2 copies are called sister chromatids. And these are joined together by a structure called a centromere. And you see there's a centromere in the middle, and that's where they cross. Humans have 23 pairs of chromosomes, of homologous chromosomes. When each chromosome in the pair is replicated, This makes a total of 92 chromosomes, right? 23 times 2 is 46. 46 times 2, 92. See where we're getting our numbers from? Okay. Okay, so dealing with interphase. And I don't want to go into too crazy of detail here because there's so much more to get into. But the whole idea here of interphase here, it sets the stage for the cell to divide. Okay? Chromosomes are first duplicated, although not visibly. Chromosomes begin to unwind tightly in a process called condensation. Okay? And again, there's a nice little video there for you to watch. You'll see what goes on here. There's your G1 phase, where the period of cell growth before DNA replication. Okay, so the cell is just increasing in size and strength. And then DNA replication happens. Okay, this is called the S phase. And then moving on into the G2 phase, this is the period after DNA replicates and the cell is preparing for division. And these are the steps involved in cellular division, finishing with cytokinesis and the finished cell, which then will continue on and do the same thing. Okay? Let me zoom out a bit here. So, there's your PMAT. As I said, when we're talking about mitosis, the cell division that follows interphase is the division of the nuclear contents known as mitosis, which is a continuous process, but it has been divided for ease of study into 4 distinct phases. PMAT, okay? Prophase, metaphase, anaphase, telophase. All right. So, what we've got going on here now. So, here we're talking about prophase. Okay, we're dealing with this section right here. And this is where the condensed chromosomes first become visible. The nuclear envelope begins to disintegrate. Okay, you'll see disintegration there, and the nucleolus disappears. Centrioles separate in the center of the cell and migrate to the opposite ends, or poles of the cell. Okay? The centrioles start to form a network of spindle fibers. There's your centriole, there's your spindle fibers up in this area here. Each cable in the spindle is made of microtubules. These microtubules grow from each pole until attached to a centromere at a disc of protein called the kinetic core. Moving on into metaphase, okay, you'll see there that things are quite different than where they were up here, right? Okay. Now you'll see there that the chromosomes are all aligned at the equator of the cell, while the centromeres are up here with the spindle fibers coming down. So the chromosome attached to the microtubules of the spindle are aligned in the center of the cell. The centromeres are aligned along the imaginary plane that divide the cell into its half. This is known as the equatorial plane. And you will see there's the electron micrograph. Okay? Detailing the exact same thing. Anaphase, now you'll see there that those spindles are pulling these stretches of DNA that were doubled to each side. Okay, and you'll see that here as well. So the sister chromatids are separated. Okay, enzymes break the cohesion and the kinetic cores. The microtubules of the spindle are dismantled, starting at the poles. This pulls the chromatids towards the poles. Okay? And telophase, you can see what's happening here now. Things are reappearing. The spindle is dismantled. A nuclear envelope forms around the new set of chromosomes at each pole. The chromosomes begin to unwind. The nucleolus reappears. And lastly, cytokinesis. okay, occurs at the end of mitosis and is a division of the cytoplasm into roughly equal halves after entering interphase. Oh, sorry, after enters interphase there. In animals, cytokinesis occurs by actin filaments contracting and pinching the cells in two. This action is evident as a cleavage furrow that appears between the daughter cells. And you'll see that there the furrow develops, and then we have two beautiful new daughter cells. Okay, there's the electron micrograph detailing the same thing. Okay, this is a great little slide here that you can just go through and have a look. Okay, make your notes from. Okay, you'll see what's happening here. There's your interphase. Okay, the whole development and doubling of the DNA. Okay, then here's your PMAT, prophase, metaphase, anaphase, telophase, in the exact same shots as before, you'll see. Okay, as well with the cartoon. The cartoon really helps explain what's going on in the micrograph, I find. And lastly there's our cytokinesis. All right. Control of cell division. All right. Cell division capacities vary greatly among cell types. Skin and blood cells divide often and continually. Neuron cells divide a specific number of times, then stop. Telomeres, they also dictate how many times a DNA can divide or multiply. And we call this there the mitotic clock. Cells divide to provide a more favorable surface area to volume relationship. Growth factors and hormones stimulate cell division. Hormones stimulate mitosis of smooth muscle cells in the uterus. Epidermal growth factors stimulates growth of new skin. Contact or density-dependent inhibition. Tumors are the consequence of a loss of cell cycle control. And we'll talk about what a tumor is, the whole idea there that, you see, you have the DNA that was in the original mother cell, okay, and we want exactly the same DNA in here and exactly the same DNA here. That's the end result of mitosis, that this and this are exact copies of this. But what happens if, when the DNA, which gets into a pretty precarious situation in DNA replication, right? This huge wonky molecule is doubling. And what are you trying to tell me, that there's no mistakes? Well, generally speaking, that's it. But sometimes mistakes can happen, where this one here gets 51%, this one here gets 49%. Now these cells are no longer working for you. This is cancer, where you have these cells that do not have your specific combination of DNA, so now these cells are working on their own. They require more food than usual, more oxygen than usual. Yes, that is cancer. Very not a good thing. All right. What do we got here now? We just got into there. So here, moving on into cancer, okay, the cell cycle is controlled by checkpoints. Okay, you remember there that there was your G1 S phase and G1 phase, right? Interphase, and there's your mitosis, PMAT, okay? You'll see there that there are checkpoints along the way, okay? Feedback from the cell determines whether the cycle switches to the next stage. 3 principal checkpoints control the cycle in eukaryotes. There's G1, G2, and M checkpoints. Okay, and you'll see there that when these cells they get checked, if they don't pass inspection, they get apoptosis, which you will know is it means programmed cell death. If something isn't cutting the mustard, it gets the chomp. Okay, that's how it works. So the G1 checkpoint determines whether the cell should divide and enter the S phase. Some cells never pass this point and are said to be in G0. Okay, they would get the chomp, apoptosis. G2 checkpoint leads to mitosis. Okay, up in here. And then we have the M checkpoint. which occurs during metaphase, okay, and triggers the exit process of the M phase and entry to the G1 phase. Okay, so it's right in here with metaphase. Okay, so there's 3 checkpoints: the G1, G2, and the M. All right. Just another shot indicating the same thing. Okay, so there's your G1 checkpoint. Here is your G2 checkpoint, and here is your M checkpoint, metaphase. Okay, just to make sure that the cell is dividing as it should be. Cancer is a growth of disordered cells. It begins when apparently normal cells grow uncontrollably and spread to other parts of the body. The result is a growing cluster of cells called a tumor. Benign tumors are surrounded by a healthy layer of cells, also known as encapsulated, and does not spread to other areas. Malignant tumors are not encapsulated and are invasive. So malignant is the worst of the two. These cells from malignant tumors leave and spread to different areas of the body to form new tumors. These cells are called metastases. Okay? So there's benign and malignant. Malignant, that's the one that's invasive and spreading. Cancer is caused by a gene disorder in somatic tissue in which damaged genes fail to properly control the cell cycle. Remember those checkpoints? If those 3 checkpoints aren't working, cancer can occur. Mutation cause damage to genes. They may result from chemical or environmental exposure, such as ultraviolet rays. Free radicals, these are the high energy molecules that we ingest through smoking, drinking, working at over barbecues. Okay, these are these high energy molecules that we ingest, and they hit the DNA and they cause trouble for the DNA. You can counter these horrible free radicals by eating blueberries, kale, leafy green vegetables, purple vegetables. They are full of what we call antioxidants. And if an antioxidant hits a free radical, it takes all of the bad stuff away from the free radical and makes it inert so it won't hurt you. Viral exposure may also alter the DNA. Absolutely, viruses can do this. There's your HPV, okay, HIV, okay, can cause cancer. 2 general classes of genes are usually involved in cancer. There's what we call the proto-oncogenes and tumor-suppressing genes. Proto-oncogenes, okay, encode proteins that stimulate cell division. Mutations to these genes can cause cells to divide excessively. When mutated, these genes become cancerous. Oncogenes. Okay, so proto-oncogenes already means that these guys here have the potential. Okay, and if they mutate, they're now oncogenes, cancer. Tumor-suppressing genes normally turn off cell division in healthy cells. When mutated, though, these genes allow uncontrolled cell division. Okay? Tumor-suppressing genes. Cancer results from damaged genes failing to control cell division. We know that. One such gene, P53, affects the G1 checkpoint. That's the first one, right? Its normal action is to detect abnormal DNA. It prevents cell division of a cell with damaged DNA until the DNA can be repaired, or it directs the cell to be destroyed if the damage cannot be fixed. If this gene becomes damaged, it will allow damaged cells to divide unchecked. And cell cycle regulation and cancer. You'll have a look here. We'll just blow this up a little bit more. Okay. And you'll see that there's your DNA. Normal P53 gene. Mutated P53 gene. And you'll see DNA mutation fixed. Okay, not fixed, and you'll see a cell divides normally, and you'll see there if everything is working fine, this cell here, which happens all the time, and you see it doesn't look good, it gets the chomp, apoptosis. But if a mutated P53 gene, you'll have a look here, these guys don't look so hot already, and now you'll see there is no checkpoint, so these guys are allowed to get out of the zoo, so to speak. Okay. Here's an electron micrograph, normal cells and cancerous cells. This is you, but it don't look like you, does it? So when a malignant tumor forms and invades nearby tissue, the following processes occur. Upregulated oncogenes, these are mutated proto-oncogenes from the previous two slides ago, or downregulated tumor suppressor genes, both cause increased cell division and tumor development. So upregulated oncogenes and downregulated tumor suppressor genes both cause increased cell division and tumor development. Cancer cells do not undergo cell death. They are immortal. Okay? So that's the whole thing. If we could somehow get a hold of that immortality and use it for good, then we could live forever, because cancer genes do. Cancer cells do. Cancer cells spread to other regions of the body. This is known as metastasis. Okay? The cells have metastasized the cancer. Cancer cells cause growth of new blood vessels so that more nutrients can be accessed by cancer genes. This is called angiogenesis. Okay, these guys here, they need lots of blood, lots of sugar, they need lots of everything, and they will command more blood vessels are built to supply them. Okay. If any of the following types of genes are mutated, cancer could develop. Genes involved in the regulation of the cell cycle, such as proto-oncogenes or tumor suppressing genes, DNA repair enzymes, genes that increase angiogenesis, making more blood vessels, genes that prevent cell death, apoptosis, genes that regulate the immune response. Okay, all of these here can have repercussions on cancer. Anti-cancer foods contain important phytochemicals that either regulate our immune response, decrease angiogenesis, or they are antioxidants that prevent free radical molecules from damaging our DNA. Why does meiosis occur? To produce gametes. Gametes are haploid, and you can see here we're starting to get into a little bit of summary. What cells undergo meiosis? Sex cells, germ cells. Okay. Oop. What is the result of meiosis in humans? Sperm and egg are the result, and ultimately a fertilized egg called a zygote. So here is your meiosis. Okay, the whole idea there of a haploid cell and a haploid cell, one coming from the father, one coming from the mother. Okay. And the whole idea, we have now created a new human being that has never before been, that DNA, that combination has never before been seen before, right? So, 23, 23, 46. Now, meiosis, very, very different, going back into the whole idea of genetic variability. Two parents can have the same, can have 4 kids, but they look all different. And there's a reason for that, and it's called crossing over. We'll get there. Asexual reproduction, no genetic exchange. Offspring have the same DNA as parents. Basically, you're making a clone. Okay, that's what asexual reproduction is. There's sexual reproduction, which is genetic variation, and gamete production via meiosis. All eukaryotic organisms have some version of meiosis. Okay? And meiosis involves 2 divisions. There is meiosis 1 and meiosis 2. DNA is replicated only before meiosis 1. Meiosis 1 separates the homologous pairs. Meiosis II separates the sister chromatids. Remember sister chromatids? Doubled of the same. When meiosis is complete, the result is that one diploid cell has become one haploid egg or 4 haploid sperm cells. Males begin to produce sperm during puberty, and this continues throughout the rest of their life. How many gametes do males produce? Millions per day. Females begin to produce eggs before their birth, and this process completes only if the egg is fertilized. Therefore, women will only have completed meiosis as many times as pregnancy has occurred. Females produce 1 egg, almost finished meiosis, per month. And you'll see here the whole idea of genetic variability, okay? And you'll see here primary sex cell, okay? And it splits. And you'll see there that what we have, all right, and these chromosomes will cross over. And you'll see here, I think it explains it better up here in meiosis 1. The whole idea here, there, that these chromosomes there, they will just, for no apparent reason, will just jump from here to here, and this one will jump here. They'll just cross over. So you'll see there that you'll have a new combination of DNA that didn't exist there before. Okay? And you'll see what's going on, the whole idea. So when you're talking meiosis here, you'll see there that there's this meiosis 1 and meiosis 2. And you'll see that that there will just, how this kind of works here is if you start here, this primary sex cell, what happens is it kind of jumps up here and does this and then kind of comes back. You'll see there that what happens is, and I'll just zoom in, and yeah, you're seeing the same, you're seeing a prophase 1, metaphase 1, anaphase, and there's a telophase as well. There are some similarities here when you're talking meiosis to the mitosis, what we just looked at. But you'll see here there that what we've got here is the chromosomes double, okay, and you have there's your sister chromatids there, and then what will happen is in prophase 1 of meiosis 1, you'll get this crossing over. And so these are new stretches of DNA that haven't been seen before, okay? And it's all about creating new, unique individuals. And then, just like in mitosis there, in metaphase there, the chromosomes there, they line up at the equatorial region there where they are separated, okay? And moving on, just trying to keep this in the same frame of the slide here, okay? And you'll see there that we have these 2 cells that are different, okay? So now let's move on to meiosis 2. And these guys here, they continue on right here. So we'll just zoom on in here. Okay, now we're into meiosis 2. Okay? And you'll see there that this is prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay? And you'll see that these guys here have now been separated. And now we kind of got to do the process again where we separate these, because these are, 46 chromosomes, and we only want 23 in each of our sperm cells or our egg cell. So what happens now, these guys here will separate again, okay? And moving on here, and you'll see one of these guys here will go into here. These guys here will go into this one. These guys here will go into this one here, and the all red will go here. So you see all red with a bit of blue. Here I have all blue, and here I have all blue with a bit of red. You have 4 different sperm cells that are completely different, and they will be passed on to the individual. Okay, that uniqueness, and it's that uniqueness that actually protects us against disease. Okay? So, there is meiosis 1 and meiosis 2 in a nutshell. Alright? Stem cells and progenitor cells. Okay, stem cells there can divide to form new stem cells. Okay, self-renewal. Okay, it's all about self-renewal. Okay, dividing to form two new stem cells can divide to form a stem cell and a progenitor cell. Okay, totipotent can give rise to every cell type. Pluripotent can give rise to a restricted number of cell types. And here, like, we're talking stem cells. If you're not familiar, the whole idea there that from a stem cell there we can get any number of different cells. And ethics aside, it's quite invigorating as far as medical procedures when you're talking about regeneration of nerve, because it's a big problem there. Somebody hurts their spinal cord, growing nerve back is very, very difficult. Okay, so these stem cells there, they give people hope. Okay, we got the progenitor cells there. These are committed cells, can divide to become any of the restricted number of cells, also pluripotent as well. We've covered this word there, apoptosis. It's programmed cell death. Another type of cell death is infarction. Now, you've heard of myocardial infarction. Okay, that's heart cell death, heart attack. That is not programmed cell death. Apoptosis is programmed. It's a natural process. Infarction is not programmed cell death. So, and now we're into a bit of review. Okay, there's your interphase mitosis and cytokinesis. There's your PMAT. Okay, here we're talking about somatic cells. Okay, body cells. There's a review for your interphase, G1, S, and G2. There's your mitosis review. I like that slide, step by step. Okay. Cytoplasmic division, also known as cytokinesis. All right. Just a little bit of review. You go back and have a look at it. Watch the video as many times as you like. Don't forget about the Q&A, the questions review that are located in the folder as well. Thank you so much for sticking around. We'll see you next week. Take care. Be well.